Good morning, everyone. Today we are talking about culture media. What is culture media? Culture media is an artificial environment which is providing natural conditions for the growth of bacteria. The artificial environment which is providing the natural conditions for the growth of bacteria. Why we want to culture bacteria? Bacteria have to be grown or cultured for them to be identified. By appropriate procedures, they have to be grown separately on culture media and obtained as pure culture for the study. The original media was used by Louis Pasteur, and there are mainly two types of media. These are liquid medium and solid medium. In the liquid medium, we will get the bacterial growth as diffuse growth. We can see the diffuse growth of bacteria in the liquid medium. And on the solid medium, we can see the discrete colonies of bacteria. These colonies are the bacterial growth. We can see discrete colonies of bacteria on the solid medium. Then the colonies are macroscopically visible collection of millions of bacteria originating from a single bacterial cell. And cooked to cut potato used by Robert Koch, it was the first solid medium. And gelatin used as the solidifying agent in the solid culture medium, but it was not satisfactory. It liquefies at 24 degrees Celsius. The universally accepted solidifying agent in culture media is agar. It was introduced by Frohese. It is used for preparing solid media. It is considered as the universally accepted solidifying agent. Agar is universally accepted solidifying agent. It is obtained from seaweeds. It does not have any nutritive value. It does not have any nutritive value, and also it is not affected by the growth of the bacteria. It melts at 98 degrees Celsius and sets at 42 degrees Celsius. And in the solid medium, the concentration of agar used is 2%. 2 to 3% of agar is used in the solid culture medium for making it the solid form. There are different types of culture media. Based on their consistency, they are of three types: solid medium, liquid medium, and semi-solid medium. Based on the constituents or the ingredients of culture media, they are of different types. These are simple medium, complex medium, synthetic or defined medium, and special media. These special media, again, of different types. These are enriched media, enrichment media, selective media, indicator media, differential media, sugar media, transport media, and media used for biochemical reactions. Based on the oxygen requirement of bacteria, media are classified into two types: aerobic media and anaerobic media. Solid media contains 2% of agar. And on the solid media, we can see the colony morphology, pigmentation property of bacteria, hemolysis also can be seen on the solid media. These are the, on the solid media, we can see the colony morphology. Different bacteria have different morphological features, and we can see pigmentation of bacteria. We can see the hemolysis or hemolytic reaction on the solid media. Examples for solid media are nutrient agar and blood agar. Then liquid media is, it does not contain agar. There is no agar present in the liquid media. Agar is the solidifying agent, and for inoculation, and this liquid media is used for inoculum preparation, blood culture, and also for the isolation of pathogen from the mixture. Example for liquid media is nutrient broth. Nutrient broth. Broth means liquid media. Then semi-solid medium contains 0.5% of agar. It is semi-solid medium, mainly used for checking the motility of bacteria. Here we can see the three types of media based on their consistency. First one is solid media on the culture petri plate. And the second one, the middle one is the liquid media. And the third, we can see three tubes. The first tube, there is non-motile bacteria. So we can see the growth of bacteria only on the stab line. And in the second and third tube, we can see the diffused growth of bacteria. That is indicating the motility of bacteria. Then simple media or the basal media, these are the media contain simple chemical composition. Examples for simple media are nutrient broth and nutrient agar. Nutrient broth consists of peptone, meat extract, and sodium chloride. Nutrient broth is broth plus 2% of agar is nutrient agar. Then complex media are the media other than basal media. They have added ingredients. It is mainly for the growth of fastidious bacteria, the bacteria which are exacting in the nutritional needs. It provides special nutrients to the fastidious bacteria. That fastidious bacteria are the bacteria which needs extra nutrients, which are exacting in the nutritional requirements. Then synthetic or defined media are the media prepared from pure chemical substances and its exact composition is known. For example, peptone water contains 1% of peptone and 0.5% of sodium chloride in water. Enriched media are the media which contains substances like blood, serum, egg. These are added to the basal medium. The substances like blood, serum, or egg are added to the basal medium. And it is used to grow bacteria which are exacting in their nutritional needs. Examples for enriched media are blood agar, chocolate agar, which contains extra nutrients for the growth of fastidious bacteria. Here you can see the blood agar and chocolate agar. Chocolate agar is heated blood agar, which is brown in color. Then enriched media are the liquid media used to isolate pathogen from a mixed culture. This media is incorporated with inhibitory substances to suppress the unwanted organism, and it is promoted only the growth of wanted bacteria only and suppresses the growth of all unwanted organism. Example, selenite F broth, which is used for the isolation of Salmonella and Shigella. Alkaline peptone water used for Vibrio cholerae. Then selective media is the media which contains inhibitory substances and it is a solid media. It's enrichment media and the selective media purpose is same. It is inhibiting the growth of other bacteria and supporting the growth of wanted bacteria only. The difference is enrichment media is a liquid media, selective media is a solid medium. Then examples for selective media are McConkey medium, which is used for the selective growth of gram-negative bacteria, thiosulfate citrate bile salt agar, which is used for the growth of Vibrio cholerae, LJ medium used for Mycobacterium tuberculosis, Wilson and Blair medium used for Salmonella typhi, potassium telluride medium used for diphtheria bacilli. Here you can see the McConkey medium and TCBS. It is used as a selective medium. Then potassium telluride media, LJ media, potassium telluride medium, selectively permitting the growth of Corynebacterium diphtheriae. LJ medium is allowing the growth of Mycobacterium tuberculosis. Then indicator media, which contains an indicator which changes its color when a bacterium grows in them. Example, blood agar, McConkey agar, and Christensen's urease medium. These are the indicator media. Here you can see the hemolytic property of the bacteria. There is a clear zone around the colony, which is indicating the hemolysis of RBCs. Here, this is the urease medium. The first tube is the original color of urease medium. And if it is a urease positive organism, it turns its color from yellow to pink. Then differential media, which is used to differentiate different types of bacteria, which has substances incorporated in it, enabling it to distinguish between the bacteria. Examples are McConkey medium, which contains peptone, lactose, agar, neutral red, and tarocolate, which is used to distinguish between lactose fermenters and non-lactose fermenters. Lactose fermenters are giving pink colored colonies. and the non-lactose fermenters are colorless colonies. The first plate is the McConkey agar, the plain McConkey medium, and in the second plate we can see two types of growth. First is the left side is the pink colored colonies, that means lactose fermenting colonies, and the second portion, the right side, is the yellow colored colonies, which is indicating non-lactose fermenting colonies. Thank you.